makmur. Menjadi tonggak dalam mencerdaskan kehidupan bangsa. Membentuk insan abdi masyarakat, pembina nusa bangsa. Universitas Padjadjaran atau UNPAD lahir dari prakarsa pemuka masyarakat Jawa Barat dan dukungan pemerintah daerah untuk memiliki perguruan tinggi yang mampu menyelenggarakan berbagai bidang keilmuan. Diresmikan oleh Presiden Soekarno pada tanggal 11 September 1957 silam. Hingga saat ini, UNPAD telah memiliki beragam capaian prestasi kelembagaan. Di antaranya, berstatus sebagai perguruan tinggi negeri badan hukum dengan akreditasi institusi A. Capaian tersebut tidak terlepas dari komitmen seluruh pemangku kepentingan. Demi tercapainya visi dan misi UNPAD, menjadi universitas unggul dalam penyelenggaraan pendidikan kelas dunia, yang mampu mendukung daya saing bangsa melalui proses pendidikan bermutu dan penelitian yang inovatif. Sarana pembelajaran yang representatif dan lokasi yang strategis di tengah kota Bandung, Kabupaten Bandung, dan kawasan pendidikan tinggi Jatinangor Sumedang menjadikan UNPAD sebagai universitas yang unggul dalam penyelenggaraan pendidikan, penelitian, dan pengabdian kepada masyarakat. Melalui program-program terbaiknya yang mampu memenuhi tuntutan masyarakat. Saat ini, UNPAD memiliki 16 fakultas dan 1 sekolah pasca sarjana dengan 152 program studi. Fakultas Hukum, Fakultas Ekonomi dan Bisnis, Fakultas Kedokteran, Fakultas Matematika dan Ilmu Pengetahuan Alam, Fakultas Pertanian, Fakultas Kedokteran Gigi, Fakultas Ilmu Sosial dan Ilmu Politik, Fakultas Ilmu Budaya, Fakultas Psikologi, Fakultas Peternakan, Fakultas Ilmu Komunikasi, Fakultas Keperawatan, Fakultas Perikanan dan Ilmu Kelautan, Fakultas Teknologi Industri Pertanian, Fakultas Farmasi, Fakultas Teknik Geologi, Sekolah Pasca Sarjana. Menyadari akan arti penting proses pembelajaran, kurikulum dan metode pembelajaran pun terus dikembangkan. Mengikuti dinamika kebutuhan para pemangku kepentingan. Saat ini, UNPAD sudah menggunakan metode pembelajaran berbasis student-centered learning, yaitu metode pembelajaran yang berbasis pada mahasiswa untuk terlibat secara aktif dalam mengembangkan pengetahuan, keterampilan, sikap dan perilaku dengan kurikulum berbasis kompetensi. Dengan dukungan staf pengajar dan tenaga kependidikan profesional, UNPAD mampu menghasilkan lulusan yang berkarakter dan berdaya saing. Sampai saat ini, tercatat puluhan ribu mahasiswa dari penjuru Indonesia dan ratusan mahasiswa mancanegara menempuh studi di Universitas Padjadjaran. Untuk mengoptimalkan proses pendidikan dan pembelajaran, UNPAD didukung oleh berbagai sarana fasilitas penunjang yang representatif. Di antaranya, perpustakaan, pusat pelayanan basic science, pusat bahasa, e-learning, rumah sakit pendidikan, serta rumah sakit gigi dan mulut. Selain itu, UNPAD juga dilengkapi dengan fasilitas pemondokan, fasilitas kesehatan, fasilitas olahraga, transportasi dalam kampus, perbankan, ruang pertemuan, dan fasilitas kewirausahaan yang semuanya terintegrasi dengan teknologi informasi. Selain mendukung peningkatan kualitas akademik, Universitas Padjadjaran juga mendukung penuh aktivitas pembentukan karakter mahasiswa melalui berbagai unit kegiatan mahasiswa. Sebagai bagian dari masyarakat, Universitas Padjadjaran menyadari betul fungsinya tidak terbatas pada pengajaran semata, melainkan mencakup aspek sosial dan budaya. Berbagai kegiatan sosial, pemberdayaan masyarakat, dan hilirisasi penelitian telah dilakukan untuk memberi manfaat nyata bagi seluruh masyarakat. Universitas Padjadjaran berdiri dan berkembang bersama masyarakat. From West Java for Indonesia to the world through SDGs. Tatar Sunda. Yang kaya akan ragam
just for the information, the event will be started very soon. Please be ready. Bismillahirrahmanirrahim. Honorable, the Dean of the Faculty of Animal Husbandry, Universitas Padjadjaran, Honorable, invited speakers, invited institutions, and all the participants, Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. On behalf of the committee, I would like to welcome all of you to the International Online Seminar with the topic Sustainable Future Livestock Production System. This is to commemorate of the 57th Disnatalis of the Faculty of Animal Husbandry, Universitas Padjadjaran, and organized by the Disnatalis Committee and the International Office of our faculty. First of all, I would like to introduce myself. My name is Diki Ramdhani, acting as a moderator in this event. The next, I would like to inform you the rundown of our seminar. So the first session will be welcoming speech and officially opening the event by the Dean of the Faculty of Animal Husbandry, Universitas Padjadjaran, Professor Husmi Yurmiati. And the second will be presentation session. The first speaker will be Dr. Ahmad Firman from Universitas Padjadjaran, will speak about sustainable succession and sustainable livestock production. And the second speaker will be Dr. Hossam Shawki from Nagoya University, Japan. He will speak about spermatogonial stem cells, a sustainable source of superior traits. The third speaker will be Dr. Mijanur Rahman from Universiti Kelantan, Malaysia. Will speak about oxalate accumulation in the tropical forage and its feeding effect on livestock. And the last will be Dr. John Gaughan from University of Queensland, Australia. Unfortunately, he is in not well condition, and his presentation slides will be distributed via a chat room. Actually, he is joining our event, as can be seen in the participant list, but he couldn't talk well and couldn't make the presentation. So everybody is welcome to download and read his presentations via chat room. So the next session, after all the speakers have presented their slides, will be discussion session, closing statements by the speakers, and last will be wrap-up session and closing ceremony of the program. So before starting to the first session, I would like to inform you our webinar rules as follows. The online seminar is free of charge. Participant audio features are automatically regulated by the host, and co-host to maintain seminar conduciveness. The presentation and discussion sessions are guided by a moderator. Questions should be sent via chat room and state any presenter to whom you are asking for. It's forbidden to use features in this Zoom seminar to advertise or commit verbal violence containing racism, sex, religion, and ethnicity elements. The results of this discussion in the form of recordings or material will be distributed only to participants who attend via WhatsApp group or email that has been registered by filling out the feedback form, and the feedback form itself, the link of that will be distributed at the end of the seminar. The material link and the absence should be shared in the chat room. Please make sure you don't miss it. Right, we are going to our first session of the event, which is welcoming speech and officially opening the event by the Dean of the Faculty of Animal Husbandry, Universitas Padjadjaran, Professor Husmi Yurmiati. To Professor Husmi Yurmiati, time is yours, please.
Okay, thank you very much, Mr. Diki. How are you, ladies and gentlemen? I hope all of you are in good condition in this difficult pandemic situation. I want to welcome everyone with my speech. Assalamualaikum warahmatullahi wabarakatuh. Dear Honorable Speaker and guests, it's with a great pleasure to welcome you to this international online seminar with the topic Sustainable Future Livestock Production System. In commemoration of 57th anniversary of Faculty of Animal Husbandry, Universitas Padjadjaran, organized by the Committee of 57 Disnatalis of Faculty of Animal Husbandry, Universitas Padjadjaran, and International Office of Faculty of Animal Husbandry, Universitas Padjadjaran. Today, we will be gaining insight from Dr. John Gaughan, Associate Professor at the School of Agriculture and Food Science, University of Queensland, Australia. Dr. Hossam Shawki, Assistant Professor in Reproductive Genetics, Nagoya City University School of Medical Science, Nagoya, Japan. Dr. Mohammad Mijanur Rahman, Senior Lecturer at Faculty of Agro-Based Industry, Universiti Malaysia, Kelantan, Jeli, Malaysia. And Dr. Ahmad Firman, Senior Lecturer at Department of Economic, Faculty of Animal Husbandry, Universitas Padjadjaran, Indonesia. And with moderator, Dr. Diki Ramdhani, he is a Senior Lecturer at Department of Animal Production, Universiti Padjadjaran. Ladies and gentlemen, the livestock sector is moving very dynamically globally. The level of demand for livestock products is highly dependent on population growth, economic growth, the level of technology use, and the availability of a country livestock population. Although the development of breeding, nutrition, and livestock health has contributed to increasing livestock production efficiency, is the future, the livestock sector will be strongly influenced by competition for the use of natural resources, especially land and water, competition between food and feed, animal welfare regulation, and socio-economic factor concerned from human health. Therefore, given the importance of this topic, Also to commemorate the 57th anniversary of the Faculty of Animal Husbandry Universitas Padjadjaran, we intend to address this issue in an online international seminar. I would like to thank the committee of this event, the committee of 57 Disnatalis of Faculty of Animal Husbandry Universitas Padjadjaran, and the International Office of Faculty of Animal Husbandry, who have been doing their best to organize this event. I would also like to thank the invited institution for joining this event from government, private, university, lecture, and all participants. Thank you so much for your participation. Lastly, I wish to thank all the speakers for their time and insight. I hope this program will extend and maintain the international network and recognition of Faculty of Animal Husbandry, Universitas Padjadjaran. Ladies and gentlemen, by saying Bismillahirrahmanirrahim, I would also like to officially open the begin this event. Ladies and gentlemen, I wish to thank you for your time to join this online seminar. I hope you can enjoy the talk and get the most out of it. Wabil taufiq walhidayah. Wassalamualaikum warahmatullahi wabarakatuh. Thank you, Mr. Diki.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Husmi Yurniati, for your valuable welcoming speech and officially opening the event. Again, thank you very much.
Thank you.
Next, we are going to our second session, which is presentation from our invited speakers. The first speaker will be maybe the organizing committee can show the curriculum vitae of Dr. Ahmad Firman, please. So while our first speaker will be Dr. Ahmad Firman, while he is preparing his slides, I would like to read his curriculum vitae. Dr. Ahmad Firman is a senior lecturer and researcher in the Department of Social and Economics, the Faculty of Animal Husbandry, Universitas Padjadjaran. He is also an expert staff in the Directorate of Food and Agriculture, Ministry of National Planning and Development, National Planning and Development Agency, or we usually call it Bappenas. He did PhD in economics from the Faculty of Animal Husbandry, Universitas Padjadjaran, and he has areas of expertise include livestock economics, agribusiness, and sociology. He has a number of publications, according to Google Scholar, about 55 publications, and his age index is 6. Dr. Ahmad Firman, are you ready with your presentation, with your slides?
Okay, thank you. Okay, thank you, Mr. Diki. I want to present, please. Okay, thank you. Thank you, Mr. Diki, as a moderator. I'm sorry to be the first presenter here because I have to replace Dr. Gaughan from Australia. I hope you are well, Dr. Gaughan. And I given this title because I worry, of course, there is no farm successor in the next future to operate the farm, because nowadays young generation views to agricultural sector has been declined. That's my opinion. Okay, next slide. Okay, the agricultural sector has been, okay, the agricultural sector has changed dramatically since the introduction of technologies, machineries, chemical use, specialization, banking access, government policy, and the emerging of industries. In developed countries, the use of technology, increased chemical use, and mechanization creates environmental problems, such as land degradation due to chemical use and air pollution due to the use of mechanization. Continuous exploitation of land without paying attention to land balance can lead to land degradation. That's why sustainable agriculture is an interesting topic to discuss, including sustainable livestock production, both nationally and internationally, because it relates to the provision of food and fiber today and also in the future. Okay, next. Sustainable livestock production or farming. Next. Okay, think about the food supply. Talking about livestock production is talking about providing food, how to serve from farm to table. Livestock production produce livestock products such as meat, eggs, and milk. Livestock products contribute to meeting the needs of animal protein for humans. Okay, Food Agriculture Organization predicts that consumption of animal foods will increase along with the increase in human population and also income. For instance, the trends in meat consumption and production in several ASEAN countries can be seen at figure 1 and figure 2. Please show the figure. Next slide. Okay. Okay. In the terms of ASEAN meat production and consumption, chicken is at the top of the supply and demand for meat compared with beef and pork. The Organization for Economic Cooperation and Development, OECD, noted that poultry meat production in 5 Southeast Asian countries, such as Indonesia, Thailand, Malaysia, Philippines, and Vietnam, reached approximately 8.04 million metric tons in 2018, with pork at 8 million metric tons, followed by beef at 3 million metric tons. You can see at figure 1. Also in figure 1, the growth of chicken consumption in ASEAN countries has reached 4.7% in the last 8 years, while beef 5.2% and pork 2.5%. In figure 2 shows overall meat production has increased since 2010 in some ASEAN countries and will continue to increase in line with the expected rise of income in the region and also human population growth. Chicken meat production was ahead of pork and beef in terms of volume as measured by tons. Unfortunately, beef production has not been able to meet the large demand for beef in ASEAN countries. So therefore, several ASEAN countries import feeder cattle or meat frozen from outside ASEAN countries, such as Australia, India, to cover the shortage of beef demand. Okay, next. Thank you. Sustainable livestock farming. Livestock production sustainability refers to the farming of animal using a system that ensures or at least favors the long-term availability of the inputs necessary to continue in operation. FAO 2017. Unsustainable livestock activity can lead to environmental damage, increased disease risk, decreased genetic variation, and consumer unhappiness. Sustainable livestock farming means a guarantee of the availability of livestock food products. However, my question is, who operates the livestock farming? Okay, again, who operates the livestock farming? There is no such word except farmers. Farmers operate farms. Without them, there is no guarantee for food supply. Therefore, sustainable livestock production is correlated with sustainable succession. Okay, next, sustainable succession. Next, farm succession. According to Bohak and Borek 2009, farm succession has been discussed and being a serious issue by scholars since 1990s in developed countries and transition countries that face structural change. The emerging of industry influenced young farmers to be more interested in working at industry than agricultural sector because of profitable activity. The derivative impact on it causes the number of farmers has declined because many young generation leave rural area to migrate to the city to find a good job. The problem is many old farmers live in rural areas and are just waiting for the time when agricultural land is sold or inherited. Okay, statistics said in Irish, the number of farmers who have aged less than 35 years had decreased by 25% in 1992 to 1999 because of the emerging industry. In Indonesia, of course, the Indonesia Statistic Bureau reported that the number of workforce who work in agricultural sector was noted approximately 36 million people in August 2017, whereas in August 2010, the number of farmers was around 42 million people, which means that the declining of farmer reached about 16% in 7 years. That's why the farm succession has been a global issue, not for only developed countries, but also for developing countries, such Indonesia. Okay, next. Okay, the view of young generation on the agricultural sector. Based on my research and the studies, there are some factors that influence the young generation views to agricultural sector. Those are no good incentive working in agricultural field, such as unpredictable climate and agricultural production. Not prestigious job because work on farm must be associated with manure and waste. And in the agricultural sector, hard efforts are required, but the results are not commensurate. Thus, the young millennial generation who are known to want to achieve quickly are not met with agricultural business, which is full of uncertainty. Ignoring smallholder producers in government policy is one of the factors inhibiting the growth of young agricultural generations from among small-scale farming families. A limited accessibility of successor to the farmland due to land conversion and ownership transfer. Okay, next. How to turn back the view of younger generation to agricultural sector? I take this sentence. Turn back crime, not crime, but turn back the view of younger generation to agricultural sector. It needs big effort to turn back young generation views about agricultural sector and do it gradually. First, promoting the agricultural sector has a high profit value compared to the sectors, to other sectors, to millennials by government, educators, parents, public figures, and so on. Two, provide the opportunity to manage livestock on an economic scale so that the farm income can be greater than the industrial sector. Make it easy for them to access financial institutions so that they can increase their business capital. Next, provide ample opportunity for them to create with digital technology 4.0 to improve business skills, business capacity, and also added economic value. Land is important to livestock farming and also agricultural sector. Therefore, the government must facilitate the need for livestock land and ensure that the land does not change into other sectors. These steps must be carried out in an integrated and sustainable manner. Okay, next. It can be concluded that sustainable livestock production means sustainable food supply, and unsustainable farm succession, unsustainable livestock production, and unsustainable agriculture. Thank you very much for my opportunity to speak here, and I give it my stage to moderator, Mr. Diki. Thank you, Mr. Diki.
Thank you very much, Dr. Ahmad Firman, for your excellent presentation about sustainable succession and sustainable livestock production. To all participants who want to ask, who want to deliver questions for Dr. Ahmad Firman, can be done through chat room, and we will prepare it for Dr. Firman to answer in the discussion session. Right now, we are moving to our second invited speaker, which is Dr. Hossam Shawki from Nagoya University, Japan. Dr. Hossam Shawki, are you with us?
Yes, I'm here.
Dr. Hossam Shawki, yes. Okay. While you are preparing for your slides, I would like to read your CV first. Dr. Hossam H. Shawki is an assistant professor at Nagoya City University School of Medical Sciences, Japan. He did a PhD in reproductive genetics, University of Fukuba School of Medical Sciences, Japan. His areas of expertise include reproductive genetics, CRISPR-Cas9, and also molecular biology. He has many number of publications, and his Google Scholar Edge Index is 5. So Dr. Hossam H. Shawki,
Yes.
are you ready for your presentations?
Yes.
Yeah. Okay, Dr. Hossam Shawki will speak about spermatogonial stem cells, a sustainable source of superior traits. Time is yours, Dr. Hossam H. Shawki.
Hello, everybody. Can you see my my presentation?
Yes, we can see it clearly.
Okay, I'm happy to join today, and thank you for the invitation. Today, I will talk about the spermatogonial stem cell as a sustainable source of superior traits. This is Hossam from Department of Comparative and Experimental Medicine, Nagoya City University. And first, I would like to give a small brief introduction about our university. We are in Nagoya City, and our university, Nagoya City University, was established in 1950 as a center for medical and pharmaceutical at first, and then developed to 7 other schools: a School of Medical Science and Pharmaceutical Science, Economy, Humanity, Design, Nursing, and Basic Science. And we have 4 campuses in Nagoya City: Takiko campus, Tanabidori campus, Sakurayama campus, and Ikita Shikuza campus. Our president is Professor Kohori Kenjori, and the aim of our university under his leadership is in which the citizen of Nagoya to feel pride and affinity. And this is our lab. We are in Department of Comparative and Experimental Medicine, and he is a Professor Oishi Sensei. He is a head of the laboratory, and also he is the director of Animal Facility Center, which is responsible for making the genetically modified animals. and they are undergraduate student and graduate student in our laboratory. And first, I will talk about the testis, which is a house of superior traits. This is a very basic things. Testis is a oval in shape, and it contains several seminiferous tubules, which is the house of sperm production. And after the sperm is produced, they move to epididymis and stored in the coda epididymis until ejaculation. And if we take one histology section from these seminiferous tubules, it contains like this. So this is the Sertoli cells inside the tubules, and it contains the undifferentiated spermatogonia, which is kind of a stem cell. And this spermatogonial stem cell can proliferate indefinitely. And after they differentiate to spermatogonia, they become no more stem cell, and they must go to development for first meiotic and second first meiosis and second meiosis to form primary spermatocyte and round spermatid, and finally the haploid sperm. And the conventional method for transmitting the superior traits, for example, if the animal is superior for milk or meat production, we usually we collect the sperm from this animal and store it in liquid nitrogen, and after that we can transfer this to the local pits. And but the problem here is that sperm carrying the superior trait are not a sustainable source. If this animal is very distinguished, it can give some, it can survive for several years, but after that we lose this strain. So we are thinking about another method for transmitting superior traits, which is using spermatogonial stem cell. So if we collected the testes from this superior animal and make enzyme digestion and collected the spermatogonial stem cell and save them in the liquid nitrogen, then we can, after that, making in vitro sperm production, and this spermatogonial is indefinitely can proliferate. And if we could make in vitro sperm production, we can transfer this sperm to local breeds. And spermatogonial stem cells are sustainable source for superior traits in that case. However, the protocol or method for in vitro sperm production from spermatogonial stem cell, no one can do so far. So we will, here I explain, so we should first know how the sperm in vivo, spermatogonial stem cell in vivo can develop, and then we can make in vitro sperm production in vitro. So Sertoli cell is responsible for this. If the Sertoli cells produce growth factor, for example, GDF9 or FGF, the spermatogonial stem cell can proliferate and become more stemless. But if they stopped producing FGF or GDF9, they produce retinoic acid instead of, and then spermatogonial stem cell can differentiate the spermatogonia and commit it to the development. So we are interested in to know. So in there is two differentiation method. First, if the spermatogonial stem cell is, only exist together with the Sertoli cells, and the second case if the all germ cells already developed. So in that case, the retinol, which is a metabolite of vitamin A, is resynthesized to retinaldehyde dehydrogenase by retinol dehydrogenase, and then retinaldehyde can synthesized by retinaldehyde dehydrogenases to form retinoic acid, and the retinoic acid finally can bind to retinoic acid receptor in an autocrine manner inside the Sertoli cells, and after that, somehow retinoic acid receptor can induce the differentiation of spermatogonium. However, how retinoic acid can induce this is unknown, because it's occur in endocrine manner, but autocrine manner, sorry, but this should be paracrine manner. So it should goes out from Sertoli cells, and this is unknown. However, after the first development for all germ cells, So there is another source for retinoic acid comes from spermatocyte, and that also unknown how spermatocyte can induce spermatogonic stem cell differentiation. So my colleague previously, he published this information, and when he knocked out, so we use a mouse as a model for understanding this method. And when he knocked out the enzymes, which is responsible for retinaldehyde dehydrogenases, ALDH1A1 to 3, specifically in Sertoli cells, you can see here the sperm is not formed. The spermatogonic stem cell cannot differentiate. However, after he injected antagonist, specifically antagonist for retinoic acid alpha, so sperm can differentiate. And in that case, he checked the genes of one of the proteins called MAFP in Sertoli cells. In this case, the MAFP cannot be expressed, but after he injected the antagonist, MAFP is highly expressed. And also he checked the promoter site of the MAFP, and he can found retinoic acid element in this promoter. And that means retinoic acid binds to the promoter of MAFP and probably regulates this. However, there is no genetically engineered mouse for MAFP at that time. So, and another research also published that about traffic jam. So traffic jam is one of the MAF family members in Drosophila. And when they mutated this traffic jam, then spermatogonic stem cell cannot differentiate to differentiated spermatogonic. And traffic jam is very highly similar to CMAF and MAFP, which is a MAF transcription factor in mammals. As you can see here, the amino acid is highly similar. So what is MAF family of transcription factors? So this family contains of a small MAF and large MAF. Small MAF contains MAF K, MAF G, and MAF F, and large MAF members is MAF A, MAF B, CMAF, and NRL. And this kind of family is a kind of transcription factor. So large MAF contains a trans-activation domain, basic domain, and the leucine zipper. But small MAF lacking the trans-activation domain. So this kind of proteins can bind to many other proteins through what is called MER site, is a known sequence in the promoter, and they can induce their transcription. So we checked first in the testes whether which one of the MAF transcription factor expressed, and we found that MAF A and MAF A, MAF B, and CMAF MAF expressed in pancreas, and other is, but only MAF B and CMAF expressed in testes. And we then collected the spermatogonic stem cell and Sertoli cells, so by FACS analysis, and to do check the expression. So we found that only, so this population represented the the germ cells, and this population represents the Sertoli cells. And if we see the Sertoli cells, we can find around 50% of these Sertoli cells express MAFP. So in this case, we used MAFP GFP knock-in mouse. And also we found around 0.4% express germ cells express MAFP. So we then localize the expression of MAFP in the testis, and we can see here by immunohistochemistry, for example, here the red is MAFP, and this is another marker for germ cells. And MAFP could be not expressed in differentiated spermatogonia, but it can express in undifferentiated spermatogonia. And KIT is a marker for differentiated, and SCB is marker for meiosis, and beta-lectin is marker for the acrosome, which is located in the head of elongated spermatid and round spermatid, and vimentin is marker for the Sertoli cells. And clearly the Sertoli cells express this protein. And then we checked because there is different stages in the during the development of germ cells, and these stages we checked the expression of MAFB, where the expression is highest in MAFB in this stage, and we found that stage 7 to stage 8 is highly expressed, and these stages are known. It is a stage for the differentiation of spermatogonia from stem cell to differentiated spermatogonia. And as you see here, we also checked using immunohistochemistry, and stages 7 to 8, here this is a Sertoli cells, the MAFB expressed transitionally in this stage, but not other stage. It means that in the same time, when we cultured the Sertoli cells and we induced the retinoic acid, the expression of MAFB increased. That means there is a very strong correlation between retinoic acid and the protein which is called MAFB. And then we generated the conditionally knockout mouse by deleting the single exon of MAFB and mated this mouse with Cree driver mouse. So in that case, in this model, if we injected a drug called tamoxifen, so the MAFB exon is deleted. And we checked, we confirmed this model by injecting tamoxifen and using mating with reporter mouse. And you can see here, if the deletion is not occur, so we can see EGFB, but if we inject tamoxifen and deletion occur, we can see TDS red. And in this case, after injection of tamoxifen, we can see TDS red. And this is a time point after around 15 days of injection, we can see deletion also by BCR. And also in immunohistochemistry, we can't detect the MAFB in the knockout mouse and also Western blotting. So we confirmed the model, and then we used this strategy by after the knockout mouse, conditional knockout mouse birth. So we inject at 6 weeks of age, we injected tamoxifen, and then we confirmed the deletion, and then we injected the busulfan drug. So this is the carcinogenic drug which can, using 20 milligram per kilogram, can kill all germ cells except spermatogonial stem cell. So in that case, only in the testis, spermatogonial stem cell exists and Sertoli cells. And then, so it takes around 1 month to kill all germ cells. And then we check to confirm after 2 months. So the stem cell started to should be differentiated again. And so we checked around 17 weeks after the injection. And in that case, we surprisingly found that this knockout mouse cannot form the differentiation. So it means if we deleted the gene in the Sertoli cells and the spermatogonial stem cell cannot differentiate as we expected, and comparing to control. And we also checked whether the spermatogonial stem cell is still exist or the busulfan might have some carcinogenic effect on the spermatogonial stem cell. But we can detect the OCT4 is a marker for this spermatogonial stem cell, and it's obviously still exists. And we afraid that also there is any defect occur in Leydig cells which produce testosterone. So we checked in the knockout mouse and the control mouse, and there is no difference. So it means the Sertoli, this phenotype comes from Sertoli cells. However, we want to check after the differentiation of all germ cells, is the same protein functionally from spermatocyte to induce the differentiation of spermatogonial stem cell. So we used this strategy. At 6 weeks of age, we injected the tamoxifen to delete the protein, and 8 weeks of age, we checked the deletion by BCR, and then at 12 weeks, so in this case, we didn't injected busulfan to kill all germ cells except spermatogonial stem cell. So at 12 weeks, we checked by histology. However, in this case, the all germ cells exist, and also the CKO has no any abnormality. And we checked sperm count and progeny number, and there's no difference. So we concluded that to initiate the first spermatogenic wave, after the synthesis of retinoic acid and binding to retinoic acid receptor A and activate it, so MAFB retinoic acid A bind to promoter of MAFB, and MAFB induce the differentiation. So, but in the maintenance of spermatogenesis stem cell after the first wave, so there is a supplement from spermatocyte, but we still don't know which factor induced from spermatocyte. So, but there is one issue here that in the Sertoli cells, these Sertoli cells and MAFB express is a transcription factor. So it's expressed in Sertoli cells and cannot go out from Sertoli cells to induce the spermatogenesis stem cell. It must work this through another secreted protein. And then we did RNA sequencing, and we confirmed the deletion of MAFB expression, and we found around 140 gene is downregulated and 110 is upregulated. And among of this protein, we checked the MER site, the location where MAFB bind in the DNA and the promoter of other genes. And we found around 13 genes has this MER site from the downregulated genes. And then we checked the signal peptide. So secreted protein usually have a signal peptide, which is a short peptide present in the N-terminal, and the majority for newly synthesized proteins that destinated towards the secretory pathway. So we found that 5 of them contains this short peptide. And we confirmed one of them, which called CXCL5 is a chemokine, and we using luciferase assay. So we cloned 1 kilobase of the promoter from this protein and 1 or 2 kilobase. And we confirmed by overexpression of the MAFB in the Sertoli cells, and we found that if we increase the expression of MAFB 0, 50, 100 nanogram, so the concentration increase, the expression increase of CXCL5 in 1 kb and 2 kb. So it means this protein MAFB directly bind to CXCL5, and this is might be a secreted protein to induce the differentiation. So in conclusion, we concluded that MAFB from Sertoli cell is required for the initiation of the first spermatogenic cycle to induce a spermatogenesis stem cell differentiation, but not the subsequent wave. And this data will help to to understand and induce the in vitro differentiation of spermatogenesis stem cell if we add to spermatogenesis stem cell the secreted protein, which might be CXCL5, then we can transit the spermatogenesis stem cell to the second differentiated, and then we can continue this differentiation in vitro until we reach the final target, which is a sperm, haploid sperm. And finally, I would like to acknowledge Padjadjaran University for invitation, and Dr. Ranga and all staff and workers in university, and also my home university, Nagoya City University, and my professor and my co-workers. And thank you very much, and welcome for any questions.
Thank you very much, Dr. Hossam Shawki, for your valuable presentation about spermatogenial stem cells, a sustainable source of superior traits, which is an advanced technology in animal reproduction. To those who want to deliver questions for Dr. Hossam Shawki, please deliver your questions through chat room, and we will discuss all the questions in the discussion session. Again, thank you very much, Dr. Hossam Shawki, and we are moving now to our third invited speaker, which is Dr. Mohammad Mijanur Rahman. Dr. Mohammad Mijanur Rahman, are you with us?
Yes, doctor. Yes.
Okay. Right. While preparing your slides, I would like to read your CV first. Dr. Mohammad Mijanur Rahman is a senior lecturer in the Faculty of Agro-Based Industry, Universiti Malaysia Kelantan, Jeli Kelantan, Malaysia. He did his PhD in agricultural science, majoring animal nutrition from Kagoshima University, Japan. His areas of expertise include grass and forage science, animal nutrition, poultry science, and also animal biotechnology. So he has a number of publications for about 71, and his age index, according to Google Scholar, is 15. Right. Dr. Mohammad Mijanur Rahman, if you are ready, time is yours then.
Okay.
So Dr. Mijanur Rahman will speak about oxalate accumulation in tropical forage and its feeding effect on the livestock. Please.
Okay, thank you, Dr. Diki Ramdhani, to introduce me. First of all, I would like to thank organizing committee of this international online seminar for giving opportunity to share my research experience. Ladies and gentlemen, Assalamu alaikum and very good morning to all. Today, I like to talk on topic about oxalate accumulation in tropical forest and its feeding effect on livestock. All of us, we know that feeding cost for livestock is about 60 to 70% of total production cost. But we can reduce the feeding cost by supplying the forest plants. But we know that oxalic acid is a common constituent of plants because it can play various roles in plants, including calcium regulation, ion balance, plant protection, tissue support, and heavy metal detoxification. So oxalic acid, it can be found in plants as soluble form and insoluble form. So what is soluble oxalate form? For example, sodium oxalate, potassium oxalate, ammonium oxalate. And also insoluble oxalate like calcium oxalate, magnesium oxalate, iron oxalate. So there are many forest plants that can sometimes accumulate oxalic acid to potentially toxic concentration in animals. For example, setaria, buffalo grass, pangolin grass, kiku grass, and also napier grass. So in our previous experiment in Japan, we have observed that what is called, we've grown the napier grass, 4 varieties, Rokuana, Markeron, Dwarf Early, and Dwarf Late. So we have observed that early summer, what is called napier grass grown in early summer, they showed a higher oxalate content, soluble oxalate, 3.77% compared to late summer and late autumn grown napier grass. However, no differences were observed among the varieties, but dwarf varieties showed comparatively at higher oxalate compared to the non-dwarf type. And if we see the plant part, we can see here leaf contain more soluble oxalate content, about 2.78% compared to the stem. So in another experiment in Kelantan, we did experiment on 7 varieties. in Napier grass. So in this experiment, we have observed that dwarf variety also contain more soluble oxalate content, about 3% compared to the non-dwarf varieties. And also similarly, leaf also contain more oxalic acid, about 3.45%. This one based on dry matter compared to other varieties. So this one is another experiment. We have grown the Napier grass with 2 levels of nitrogen fertilizer, 5 gram nitrogen per meter square per year. Another treatment, 30 gram nitrogen per meter square per year. And we harvested the Napier grass at different cutting interval, like 2 weeks, 4 weeks interval, and 8 weeks. So we have seen that soluble oxalate content declined as the harvest interval increased. So in this experiment, we can recommend that it is inadvisable to feed large quantities of young Napier grass to ruminants. So this is another experiment we have conducted under high level of nitrogen fertilizer as a urea fertilizer. We have seen like 0 gram, 10 gram, 30 gram, 60 gram, and 90 gram nitrogen per meter square as urea. So we have observed that soluble oxalate content in the first and second cut forest we are not affected by the level of nitrogen application. However, soluble oxalate content in the third cut forest tended to decrease with increasing level of nitrogen level. So figure 3 shows that insoluble oxalate content in Napier grass as affected by nitrogen fertilization. So similar to soluble oxalate content, insoluble oxalate content in the first and second cut forest were not affected by the level of nitrogen application. But insoluble oxalate content in the third cut forest appeared to increase with increasing nitrogen level. And this is data is from another experiment. We grown the Napier grass, dwarf Napier grass under hydroponic condition, and we give nitrate fertilizer, and another treatment is ammonia fertilizer. So we can see here nitrate nutrition showed relatively higher contents of soluble oxalate, insoluble oxalate, and also total oxalate content in the upper ground part of the Napier grass, as well as in the root than the ammonium nutrition, except for insoluble oxalate content in the root. So from this experiment, we can conclude that application of nitrate fertilizer as a sole source of nitrogen should be avoided during oxalate-rich forest cultivation to prevent elevated levels of oxalate. So this table 3 shows the mineral content in Napier grass. So we can see here potassium concentration in plant tissue appear to decrease with the increasing level of nitrogen fertilizer, whereas calcium content in the plant tissue appear to increase. So tended to increase with increasing level of nitrogen fertilizer. However, no differences were observed among what is called the sodium and magnesium concentration among the treatments. So, and then table 4, we can see here the nitrate nutrition exhibited or resulted relatively higher concentration of calcium, magnesium, potassium, and sodium in the upper ground part, as well as in the root than the ammonium nutrition. So except for potassium concentration in the root. And also we conducted another one experiment. We tested about the salinity stress on oxalate content in Napier grass. So in this experiment, we conducted 5 treatment, 0 millimole, 0, 100, 300, 600, and 900 millimole sodium chloride. And from this experiment, we can conclude that 100 millimole sodium chloride resulted a higher soluble oxalate content, but the differences were not significant. So results indicated that where the soil is high in sodium chloride, Napier grass will tend to grow well and also be low in oxalate. Means that salinity is not problem for Napier grass production in terms of oxalate content. So in this figure, I want to show the relationship between soluble oxalate content and potassium concentration in plant tissue, and also sodium concentration in plant tissue. So soluble oxalate content in plants was highly correlated with potassium content, while no relationship was found with the sodium content. So this figure shows the relationship between insoluble oxalate, calcium and magnesium content in Napier grass. So here also we can see insoluble oxalate content was highly correlated with calcium and magnesium concentration in plant tissue. So however, no relationship was found between total oxalate content and nitrogen as urea. So when we applied urea fertilizer, no relationship was found between oxalate and nitrogen content in plant tissue. So now I am going to talk a little bit about the feeding effect of oxalate on livestock. So in this figure, what is the fate of oxalic acid in the digestive tract? So when a ruminant animal consume an oxalic containing plant or forest, the oxalate is metabolized in 4 possible ways. There are 4 ways they can do metabolize. So first, when soluble oxalate intake by the animal, they can be break down in the rumen with the help of rumen bacteria, Oxalobacter formigens, and then they can produce harmless formic acid and carbon dioxide. And secondly, when calcium is taken with dietary feed that is high in soluble oxalate ion in the rumen or intestine combined with calcium or magnesium to form insoluble oxalate crystal, which are then eliminated in the feces. This form of oxalate cannot be absorbed in the body. That's why animals show the calcium deficiency and mineral imbalances in their body. And thirdly, when calcium is low in the diet, soluble oxalate remains soluble in the liquid portion of the intestine contents and is readily absorbed from the intestine into the bloodstream. So if the oxalate ion concentration become very high in the blood, being filtered by the kidney, so it may combine with calcium or magnesium ion to form insoluble oxalate crystal. So that may block urine flow and cause the kidney failure. And also, as a result, reduce the serum calcium level and also renal failure. So the last one, the fourthly, insoluble oxalate from ingested plants may pass through the digestive tract without any harmful effect on the body's metabolism. So this one is called calcium unavailability by the animals. So there are many factors that could influence the susceptibility of animals to oxalate poisoning, such as chemical form of the oxalate. So what kind of chemical form are available in the plants, that one can affect on the susceptibility. For example, soluble oxalate complexes with dietary calcium and forms insoluble calcium oxalate. This leads to disturbances in calcium and phosphorus metabolism involving excessive mobilization of bone mineral. And number 2, age of the animal. Young animals are generally more susceptible to toxicity than adults. And number 3, adaptation of animals to oxalate-rich forests. So hungry and unadapted ruminants are the ones most susceptible to oxalate toxicity. If ruminants are slowly exposed to a diet in oxalate over an approximately 4-day period, the population of oxalate degrading bacteria in the rumen will increase to a level sufficient to prevent oxalate poisoning. The ruminants adapted to diets with high oxalate content can tolerate oxalate levels that may be toxic or lethal to unadapted ruminants. And also composition of the diet also can affect on the susceptibility of animals to oxalate toxicity and availability of water for animals. So to prevent oxalate toxicity, we need to ensure animal can get the water all the time. So if thirsty, so it will affect on oxalate toxicity. So this table shows the blood calcium level of animal consuming grasses with different levels of oxalic acid. We can see here one researcher, Weinstein et al. 2004, they observed that high oxalate containing Napier grass, if cattle fed high oxalate containing Napier grass, they showed low blood calcium level, means lower blood calcium level compared to those fed low oxalate containing Napier grass. We can see here 5.8 and low oxalate containing grass 6.8 blood calcium level. Similarly, in the goat also same. So goat fed high oxalate containing Napier grass, they showed only 8.1 milligram per dilution, what is called compared to 8.5. And sheep also, in our previous experiment, we have observed that high oxalate containing grass, they showed low blood calcium level compared to those fed low oxalate containing Napier grass. So here I want to say those what is animal fed grass, if have high oxalate containing grass, they showed low blood calcium level, means a lower level of blood calcium level compared to grass containing low oxalate grass, low oxalic acid. So this is the our and other people's the data. So here the critical levels reported in the literature for oxalate in animals are summarized in this table. So we can see here this level is the critical level. So this is the soluble oxalate level in the forest. So this one is the incidence and this one is the references. So we can see here like 3.01% soluble oxalate in Napier grass. So buffalo calf showed toxicity. Here another 2% or more soluble oxalate in ruminants, acute toxicity. So another one like toxicity in cattle and buffalo calves if animal fed 3.01% soluble oxalate. And the last one is like 2.66 or 2.75% soluble oxalate in the feed, reduced serum calcium level in cattle and goat. So I want to make conclusion now, like based on our previous findings, we can conclude that oxalate content in forest can be varied according to season, forest species, botanical fraction, cutting interval and fertilizer application. So it is inadvisable to feed large quantities of young Napier grass to ruminants. Applying excess levels of nitrate and potassium fertilizer should be avoided. Applying calcium fertilizer may be encouraged to reduce soluble oxalate. So dietary oxalate may result in oxalate poisoning in domestic animals. So limiting oxalate ingestion to less than 2% soluble oxalate would be appropriate for protecting the ruminants from oxalate poisoning, even though blood calcium level may decrease. In case of non-ruminants, levels of less than 0.5% soluble oxalate in their diet may be acceptable. Feeding large quantities of oxalate-rich forest to ruminants is inadvisable unless the animals are gradually introduced to the forest to allow the build-up of oxalate-degrading bacteria. Horses should not be fed a diet of predominantly oxalate-rich forest as nutritional disorders can result. Animals should not be allowed to become hungry or thirsty while feeding or grazing on oxalate-rich plants. So supplementary dicalcium phosphate in the diet before and during high-risk oxalate exposure is an effective means of reducing losses. High levels of dietary calcium bind with oxalate in the rumen. Insoluble, non-absorbable calcium oxalate, animals may be provided calcium in a salt mixer. So feeding of calcium fertilized grass can minimize the negative effect of oxalate. Diets can also be supplemented with other low oxalate containing plants to help reduce the overall intake of oxalate containing plants. Meaning that we need to give other kind of forests, low oxalate containing forests. So we cannot depend only Napier grass or only Setaria. So we need to mix with other feed ingredients. Soaking in the feed in water and then straining of the water can also reduce the oxalate content of the feed and help to reduce its toxicity. Silage making may be another option to reduce oxalate content in forest plants. So adequate water intake is also necessary to prevent the formation of kidney stone. So thank you very much. So this is the end of my presentation. So if you have any question, please answer or send to my email. Thank you.
Thank you very much, Dr. Mohammad Mijanur Rahman, for your excellent presentation about oxalate accumulation in our tropical forages and its effect on the livestock. Right, those who have questions to be delivered to Dr. Mohammad Mijanur Rahman, please do it through a chat room, and we will discuss all the questions in our discussion session after this.
Okay, Doctor. Thank you.
Again, thank you, Dr. Mohammad Mijanur Rahman, for your interesting presentation.
Thank you.
Right. Actually, Dr. John Gaughan is joining us here, but again, I would like to inform you that he is not in well condition. So he has sent us his slide material, and we will distribute his material on the chat room. The committee, please distribute Dr. John Gaughan's slide material so that participants can download it and read it. So in this opportunity, I would also to read Dr. John Gaughan curriculum vitae. So Dr. John Gaughan is an associate professor in School of Agriculture and Food Sciences, the Faculty of Science, the University of Queensland, Australia. He has affiliated as associate professor at the Centre for Animal Science, Queensland Alliance for Agriculture and Food Innovation. He has areas of expertise to include heat stress, heat load, climate change adaptation, thermal biology, and also ruminants, both cattle and sheep. So he has so many publications, 225, and his Google Scholar Edge Index is 35. So all participants, again, who are interested to see Dr. John Gaughan's slides, please download it from our chat room. So if you have questions to Dr. John Gaughan, again, you can write your questions via chat room. But if he is not able to respond, then you can email him, and his email address is available in the opening of his slides. Thank you. Right. Now we are going to our next session, which is a discussion sessions. There will be several parts of discussion sessions, and the next one will be our first part of discussion sessions. Hoping that the committee will prepare questions and share on the screen, please. Dr. Gaughan actually gave his resume of his presentation on the chat. Please be informed on the chat.
Sorry, Mr. Dicky.
Yes.
Have to answer the question first, or wait until the question all in the chat done?
Yes. I will read first the question, so all participants can be informed the questions. Then you are after that you can answer the question. So the first question is going to you, Dr. Ahmad Firman, and the question is: the Faculty of Animal Husbandry to motivate and convincing the millennial generations that work in agricultural sector has a high profit. What the faculty has a specific program? Thank you. This question is actually from from Miss Ati R from BKPP West Java. So you can respond to the questions, Dr. Ahmad Firman, please.
Okay, thank you, Dr. Dicky. Thank you for your question, Bu Ati from the KPP Jawa Barat or West Java. I think it is interesting question. In my experience to lecture my student, I just to give them the best lesson, not only in theoretical concept, but also in the implementation of the theory in every courses. For instance, I invite speaker from the success millennial who raise livestock in the farm to talk in the front of the class to motivate the student that livestock business have a good opportunity to be a rich man. That's why I try to invite many speakers who have success in the farm. I invite in the class to join the class, to share the information, to share his experience to raise the livestock and getting a rich man. So that's why how to that activities that I want to share to them because I want to my student to show that this livestock business have a good opportunity to livelihood for in the future. That's my question. Thank you.
Right. Thank you very much for your answer.
Okay. Oh, sorry. What the faculty has a specific program? There is no such as a specific program. Just maybe in the one of course there is entrepreneurs courses. That's maybe I have to underline the course because that the course is the specific program to enhance or to motivate the student to be an entrepreneur in livestock business. Thank you.
Right. Thank you for your answer, Dr. Ahmad Firman. Hoping that Bu Ati is happy to your answer.
Okay, thank
right.
you.
The next questions will go to Dr. Hossam Shawki. I read the questions. I'm sorry if my question might be far from your topic, but I'm wondering about spermatogonial stem cells advantages. As we already know that embryonic stem cell is popular in stem cell technology, and SSC, in my opinion, might be another alternative technology for stem cell. However, is there any added value from SSC technology compared to embryonic stem cell? And it's reliable to be applied in animal livestock in developing countries with their limitations. So this question is from Dr. Rangga Setiawan from Universitas Padjadjaran. Dr. Hossam Shawki, please, could you respond to this question, please?
Thank you very much for nice question, Dr. Ranga. And your question is whether if we can get sperm from stem cell. This is what I understand. And in this case, if we want to get sperm from stem cell, because we easily can collect sperm ES cells from embryo, it's a very long way and very complicated because we have first to direct it or differentiate the stem cell to primordial germ cells, and then from primordial germ cells to spermatogonial stem cell, and then from spermatogonial stem cell to sperm. So we must pass by spermatogonial stem cell at the end. And this is a very complicated way because epigenetics play several roles, for example, to distinguish the primordial germ cells from ES cells, from stem cells. And another thing is that we want also to this, and also we can collect the stem cell from embryo. So, and we want to first collect the spermatogonial stem cell from adult sire, which have the nice traits and distinguished traits for milk and meat production. And we have to check, make several checks for and test for this sire first. And then we collect it from the best sire, the spermatogonial stem cell. So we shorten the time from making the stem cell to sperm finally. And we want to make it in an easy way. And your second part is this valuable for or reliable to do in livestock? So far, if we could make the nice protocol and efficient protocol to differentiate the spermatogonial stem cell to sperm, it's become very powerful because spermatogonial stem cell can proliferate indefinitely, and we very easily to differentiate in the laboratory and increase so much. So the problem here is to differentiate them to final target, which is a haploid sperm. And if we can differentiate so many spermatogonial stem cells, then we can easily transfer this sperm to anywhere. The same as a conventional way now. We stock the sperm and send them in liquid nitrogen to anywhere. Thank you for your question. I hope this is answered your question.
Thank you for the answer. Hoping that Dr. Rangga is happy with the answer. Right. We are moving to the next question, please. Right. This question goes to Dr. Mijanur, Mohammad Mijanur Rahman. The question is, soaking feed in water can reduce oxalate content. Do you have any specific technical details on how long the soaking process and what is the water temperature? So this question is from Mr. Rifki Ismiroj. Dr. Muhammad Mijanur Rahman, could you respond to this question, please?
Yes, thank you very much. So regarding this question, I think the water temperature, just normal temperature. There is no specific any temperature. So we just need to soak in water about 2 hours is enough, I think, 2 hours or 3 hours. So in our experiment showed that what is called soluble oxalate, like sodium oxalate, potassium oxalate, ammonium oxalate. So if we soak inside the water, so that soluble, because this is soluble, so if we remove the water, then it will reduce the soluble oxalic acid. So specific technical details. So not any specific technical details. So just chop the grass, but we need to chop, means cut into small pieces, and we need to soak inside the water. So we just what is called wash, and then we need to drain out the water, and the remaining the grass we can give to the animal. So it can reduce the soluble oxalate about 60 to 70%. I think I got the answer. I think got the answer.
Yeah. Hoping that Mr. Rifki Smiraj will get the answer.
Yeah, so it is not complicated. The soak in the water, and then
Right.
that water you need to throw it.
Right. Thank you for your response, Dr. Muhammad Mijanur Rahman. To Mr. Rifki Ismiraj, if you have still any curiosity, you can still write it on the chat room.
I want to interrupt here.
Yep.
As I mentioned in my presentation, a plant contains 2 types of oxalate: soluble oxalate and insoluble oxalate. So soluble oxalate is harmful in the body, animal body. Insoluble oxalate not harmful. So soluble oxalate, if we soak in the water, so a significant amount of soluble oxalate we can reduce.
Okay, that's good information then. Okay, again, thank you, Dr. Muhammad Mijanur Rahman, for your response to the question. Right, now we are going to the second Q&A session. The committee, could you please share the question, please? Right, there is a question from Dr. John Gaughan to Dr. Ahmad Firman. The question is, succession planning is also an issue in Australia. So does Indonesia have any government programs in place to address this issue? And also, what do you think can be done to bring your young people back into agriculture, including livestock sector? Could you respond to these questions, Dr. Firman?
Okay, thank you. Thank you, Mr. Diki. Well, this question is interesting and it is hard to question, of course. But, well, Indonesia have a program like to enhance youth generation to drive the millennials to be to address agricultural sector. In this government, such as Agriculture Ministry, have the millennials program that support to millennial to be entrepreneur, agricultural entrepreneur. That the program is like PWMP. That I part of the program, and also the program support by Agriculture Ministry to have a chance to drive to enhance the business in the agriculture sector, such as livestock, create the business to be the business, and then the government support the capital, only 35 million per group, and then we deliver it the finance, the program, and also to facilitate them how to be a good entrepreneur business. That's the program in this Ministry of Agriculture. And what the program can bring your the young generation back to the agriculture, I don't have any guarantee about that, because many you know that the millennials have a view to be a quick healed, to be a well person. So the agricultural sector need effort to build the business and also the time, because not only and also patient, I think, because we cannot say that the agriculture sector have a good what do you mean that have a good livelihood, but also how to young generation to support, to have strong, to build the business without what we can say, oh, I give up. Just I just support my student. How to I just support my student and motivate my student to be a business, not to be an employee, but to be the entrepreneur is have more payment, more have good income than employee. So the livestock business is a part of entrepreneur business. That's, I don't have a guarantee about that, the young generation. But in my experience, in cooperative with the PT Frisian Black and KPBS, there is the program such as Young Generation Program to train the young generation how to raise the dairy cattle. There were 2,000 participants, but the end of the program just only 8 persons survived in that program. So yeah, it is depend on the young generation itself. Thank you very much.
Okay, thank you, Dr. Ahmad Firman, for your answering the question. Right. The next question, please. So this question goes to Dr. Mohammad Mijanur Rahman, and the questions is about how to improve mineral and nutrition of grass if it's planted in gambut land. You know a gambut land?
No,
I think
Doctor.
Bahasa Indonesia or Malay term. Yeah, this is from Dr. Saulan Sinaga from Universitas Padjadjaran. Can you answer these questions, Dr. Muhammad Mijanur Rahman, please?
Yes, Doctor. So may I know what is gambut land?
Gambut land is the land which is, if I'm not mistaken, it's high in humidity.
Oh, high in humidity.
Or
I'm sorry,
Dr. Liza. Yeah, Liza, can you explain, because Dr. Liza is the expert of grass science, please.
I'm sorry, Dr. Diki. Gambut land is a peatland, Dr. Mizan. Peatland.
Do you have any definition of that land, Dr. Liza? Oh, peatland, yeah.
Yes, Dr. Gaughan, that's true, peatland.
Okay. So in general, I want to say here, to improve the mineral and nutrition in grass, so we need to what is called apply the manure, animal manure, what is called like cow dung. cow dung or poultry litter. So animal manure we need to apply to the soil. So we cannot expect from the plant without giving any fertilizer. So we need to give appropriate amount of fertilizer according to the local recommended dose. We need to give the nitrogen fertilizer, potassium fertilizer, then what is called phosphorus fertilizer. So and we need to give also calcium fertilizer. So, but before giving calcium fertilizer, we need to check the soil pH about the acidic soil or what is called the alkaline soil. So, means I want to suggest here we need to give animal manure, the cow dung, poultry litter, like that, and also we need to give some chemical fertilizer, like as I mentioned, urea and muriate of potash, and also the phosphorus fertilizer. Then we can increase the nutrition in the plant.
Okay.
So I think got the point.
Yeah, Dr. Muhammad Mijanur Rahman, how about oxalate content? Usually in grasses grown in peatland, is it high or low?
It means about the effect of soil, not much research has been done yet. The soil, so no data is available about the soil.
Okay. Okay, that's fine. Thank you for your response to the questions. And we are going to the next questions, please. Right. This questions goes to Dr. Hossam Shawki. The questions is about how about the level of abnormality of spermatogonial stem cells or SSC. Would you like to explain the fertility to be resulted from this SSC? This questions from Dr. Nur Khalidah Solihati from Universitas Padjadjaran.
Thank you very much for your question. To answer this question is very difficult because until now we cannot differentiate the spermatogonial stem cell in vitro and to check their efficiency or the abnormality. But we can differentiate spermatogonial in another way by transplantation of spermatogonial to another testis, or making xenography under the skin of another animal. And this method, the successful rate is around 12% if we transplant. And the produced animal or the born animal looks normal, and it's epigenetic. If they make, some people make a epigenetic check and it looks normal. And I hope in near future we can, if we induce the differentiation of spermatogonium in vitro, then we will check the efficiency or abnormality of the sperm produced from this method. And we hope to reach the best method in near future. Thank you very much for your question. I hope this answers your question.
Okay, thank you, Dr. Hossam Shawki, for your answer. Next question, please. So this questions again goes to Dr. Muhammad Mijanur Rahman. It's from Dr. Nopi Mayasari from Universitas Padjadjaran. The question is, could you give some solution regarding hypocalcemia in early lactation in dairy cow related to oxalate acid in tropical forage? So do you have any idea to respond these questions, please?
Yes. So, okay, thank you very much, Mr. Nobi Mayasari, for asking good question. So usually the dairy cow who are during the pregnancy, the animal need more calcium for their pregnancy, for growth or development of the fetus. So in this time, we need to give enough amount of calcium to the animal. So I suggest during this time, during pregnancy period, need to give the dicalcium phosphate. Dicalcium phosphate, or we can give UMMB, urea molasses multinutrient block. Or also we cannot depend only one source of grass. For example, we cannot give Napier grass 100% to the cow. So we need to give mixtures of different type of grass. So as a result, we can reduce the amount of oxalate. And I think that's all. Yes. So we need to give the calcium source feed ingredient to the animal.
Thank
And
you.
we need to, we need to give a mixture of different grasses.
Okay,
So
thank you.
Please
for
don't depend on. Okay, doctor.
Dr. Nopi, do you have any next question, curiosity about?
No, I think.
question?
Yeah, thank you very much for the answer. Actually I already got the direct answer from Dr. Mijanur to me, so thank you very much. It's very interesting. I think we have some ideas actually to have join research collaboration. Maybe I will contact you in the future. Is it possible?
Yes, possible, doctor. Yes.
Thank you so much. Yeah, thank you, Pak.
Thank you, Dr. Nopi, for your question. Right, we still have time. Can the committee share the next question, please? So this question is again to you, Dr. Mijanur Rahman. The question is, how does the process of making silage affect the oxalate content in the forage plants? This is from Lita Nur Soleh from Universitas Padjadjaran.
Okay, thank you very much for making question, Lita Nur Soleh. I want to say here, because if we make silage, so this is under anaerobic condition, means during fermentation, inside the silage have a lot of billion of microorganisms. So as I said, have some bacteria can break down the oxalate, so they can use as a source of energy for them. So during fermentation process, some bacteria can break down the oxalic acid, but they cannot break down 100%. But at the end, what is called the amount of oxalate can be decreased. And that amount is safe for animal feeding. As I said in my presentation, in animal diet, for example, ruminant, 2% soluble oxalate is acceptable in the diet, but we cannot give more than 2% soluble oxalate in their diet based on dry matter. So if we make silage, usually the amount of oxalic acid can be decreased less than 2%, and it is not harmful to the ruminants. I think it got answered.
Right. Thank you very much for the answer. Hoping that Ibu Lita Nur Soleh is happy with the answer. Right, we still have time actually, and we still have several questions. And this one, the next questions is also from Dr. Iin Susilawati, and this question goes to you again, Dr. Mohammad Mijanur Rahman. The question is, which is the more dangerous effect on livestock? Insoluble oxalate or soluble oxalate?
So thank you very much for making this question. So as I said in my lecture, there are two types of oxalic acid, oxalate salt, insoluble oxalate and insoluble oxalate. So I want to say here, soluble oxalate is more dangerous because when animal consume soluble oxalate, so sodium or potassium ion, they can be separated inside the body, and that oxalate ion can combine with the bone calcium or blood calcium and to form insoluble calcium oxalate. So as a result, reduce the calcium level in their body. So animal shows the calcium deficiency. But if animal intake insoluble oxalate, so that one is insoluble in the body. So just what is called consume, and that insoluble oxalate will come out through the feces. So no more use in the body. So this one called indirect effect, because that calcium or magnesium animal cannot utilize. So insoluble oxalate not harmful in the body, but animal are not getting calcium and magnesium. But soluble oxalate, that one is directly harmful because it is combining with the blood calcium or blood magnesium. So animal shows the shortage of calcium or magnesium in their body. So my answer is soluble oxalate is harmful.
Okay.
Thank
You for your answer, Dr. Iin Susilowati. If you have any clarifications, you can just talk in this Zoom, or you are happy so far with the answer.
Thank you, Dr. Mijan.
Yes, please, Dr. Iin Susilowati. Can you unmute your?
Thank you for your question. It means if two grass species contain the same total oxalate, but its insoluble and soluble content are different, the effect on livestock will be different. Maybe it means. So you should be we must know how must know soluble content oxalate.
Yeah, yes, doctor. So we must know the soluble oxalate content in the grass. Yes. So it is recommended until 2% in the diet based on dry matter. So if more than 2%, it will harmful to the animal. Either reduce the calcium or magnesium level in the blood, or maybe sometimes kidney stone, kidney failure, and then animal die.
Important to analyze soluble oxalate, yeah.
Yes, it is very important. So it is important to know the soluble oxalate content in the grass. Yes.
Thank you, Dr. Mijan.
Thank you, Dr. Iin Susilowati, for your questions, and thank you to Dr. Mohammad Mijanur Rahman for your answer. Before going to Dr. Ahmad Firman to answer the next questions, actually, I have questions to Dr. Hossam Shawki. Dr. Hossam Shawki, are you still with us? Actually, I'm not
here.
Yeah. Actually, I'm not an expert in animal reproduction, but it's my curiosity. You are now researching very advanced technology in animal production, but have you ever applied this advanced technology practically at farm level, and how the cost resulted in this advanced technology? Is it economically feasible at the farm level? That's my curiosity. If you could
can you repeat one more time? I can't hear clearly.
Yeah. This advanced technology in animal reproduction, such as the use of spermatogenital stem cells, is laborious. Do you think this can be applied in practically at the farm level?
Yes.
Farm level. And is it economically feasible in the future?
Yes. So far, nowadays we cannot say it's applicable for farm animal or not, because we still are searching in this area. But we we hope this becomes available for in local farms in near future, and we working so hard. Many researchers are working so hard to do this. And about the costs, nowadays the costs is very high, because we still are establishing the method. But after establishing this method, and it's become very good for transfers to spermatogenital stem cell to sperm and applicable for farm animals, then we have to think about the cost. And working on reducing the cost, reducing until reaching the minimal cost for spreading the superior genetically merged everywhere.
Okay, lovely. Thank you for your answer. Hoping that success with your research about this advanced technology and in animal reproduction.
We hope
right.
So.
We are going to the next questions. This questions goes to Dr. Ahmad Firman from Dr. Siti Darojah from Universitas Padjadjaran. Dr. Ahmad Firman, are you still with us?
Oh, yes.
Yeah. The question is, how about the policy of Ministry of Education and Culture to increase and develop the senior high school for specialists for agriculture like SMK Pertanian with scholarship to the young students? Maybe could be increase the interest them to work in agricultural sector. Could you please answer these questions?
Okay, thank you, Mr. Diki. What I said before to Dr. Gaughan, that the government supports agricultural education with building some agricultural advocacy and a special school for agriculture. But what we said that for instance, however, when PT President Black collaborated with the KPBS, what I said before, or KPBS is milk cooperative to facilitate them in the program, young generation program, from the 200 youth participants followed by this program. At the end, the program just only 2% survive in this program. So that's I think the participant is all most of them graduation from senior high school and also agricultural school. So what I said before, that millennials have own views to see the agriculture. They want to something quick yield, to be a well-paid person, whereas the effort to build the agricultural business need time and patience. That's a big effort to turn back view to agricultural sector for a young generation. It is not for only parents to support them, but also all stakeholders, including government, educators, legislative, and executive legislation, to drive your generation to work in agriculture. Perhaps maybe provide them an opportunity to create their business in agriculture with their idea, with their skills. I think let them to create something. Maybe they can find a new business in livestock, new livestock business. I think with the, just like with the only use the phone, the young generation can do that with the in life in hand, like now in happen. Just let them to be a creative person. That's all. Thank you very much.
Okay, thank you very much for your answer. I think the next will be our last questions in the discussion session. Please, the committee, can you share? This question is still for you, Dr. Ahmad Firman. What is the role of the government in fulfilling sustainable livestock production in Indonesia, apart from depending on the import sector? This is from Bagja Nugraha. Please, could you respond to these questions?
Okay, this is the last question, I think, but many questions in the chat. Okay, thank you for your question, Mr. Bagja. Okay, the government of Indonesia, via Directorate General Livestock and Animal Health, has distributed many programs to support livestock sector in Indonesia. But unfortunately, the food demand of animal products, especially beef meat, does not have an equal quantity with the beef domestic production. That's why maybe if not, I'm not mistaken, 1.3 million feeder cattle import from Australia per year to Indonesia. So there is no option to import such as product. But I really appreciate it that when Directorate General Livestock and Animal Health have launched what we call the SIWA program, what we call in English special program in it's very hard to say in English, but SIWAB program to implement artificial insemination massively with support feed and feed program, health program, and service program since 2007. In my opinion, this program can be reduced the beef imported gradually. So I hope government do programs such as SIWAP again to increase beef cattle population to make balanced supply and demand of beef meat. That's my last answer. Thank you very much.
Okay, thank you for your answer, Dr. Ahmad Firman. Right, before I wrap up this session, I would like each of the invited speakers have their closing statements. Could you be the first, Dr. Ahmad Firman, to have a closing statement?
Okay, wait a moment. What kind of closing? Okay, I believe that sustainable livestock production must be included the succession. Sustainable succession. Without farmer, we can operate the farm. Without farmer, we can operate the machine, mechanization, something like that. Without farm, there is no food supply again. That's it. Thank you very much.
Okay, thank you for an excellent short closing statement. Now, Dr. Hossam Shawki, could you have a closing statement about your topic of your presentation then, please?
Thank you very much. I would like to acknowledge Padjadjaran University first for invitation, and our technology, we hope that we could in near future produce spermatogenetic stem cell differentiation to sperm, which will decrease the costs of maintaining the sires. And for example, if you keep the sire, it costs a lot of money to feed them. And we can use a sustainable way for producing sperm from distinguished animal in near future. Also, our target, this technology also will help also in medical science, which we are a medical school. For example, for people who has oligospermia, which has no sperm due to several mutations in the Sertoli cells or genes. And this technology will help to understand why they cannot produce sperm as well in vitro if we succeeded. So we hope to succeed soon to help both medical and also at a farm, livestock farmers. Thank you very much.
Okay, we hope so. Thank you very much for your closing statement. And now we are going to the third closing statement from Dr. Mohammad Mijanur Rahman. Please.
Yes, Doctor. First of all, I'd like to thank University Padjadjaran for organizing this seminar. So my statement I want to say here, whatever we are doing, experiment or research, so that research data or result, we need to bring to the farmers level. So we, based on my experience, there are many what is called invention or what is called technology, but farmers they don't use it. So we need to work together with the farmers using our data. Okay, thank you.
Okay, thank you very much. All the participants, please give applause to all of our invited speakers then for their valuable, interesting presentation and discussion. Right, I will move to wrap up a session. I have small conclusion actually. It's that sustainable future livestock production system is affected not only by one sector but also many sectors related to farm animal livestock sectors. The first one is, as mentioned by Dr. Ahmad Firman, who have quite problem where young generations are likely to have less interest to work in agriculture, including livestock sector. Here, we need to encourage them to love agriculture. Otherwise, we will not have massive young generation to continue to improve our livestock sector to become sustainable in the future. The second one, advanced technology in animal reproduction is also able to improve our sustainable future livestock production system. As mentioned by Dr. Hossam Shawki, that advanced technology like spermatogenial stem cells to improve can, although so far it's still applied in medical science to humans, but hoping that the future it can be used for livestock production sectors. So hopefully in the near future, this technology will be practically and economically feasible in the near future. And the third, the issue of sustainable future livestock production system is also about animal nutrition. So here, nutritionists have to formulate the diet carefully. For example, oxalate, as mentioned by Dr. Mohammad Mijanur Rahman, that oxalate accumulations, especially the insoluble one in the grass or in other feed materials, if the intake is higher than its requirement, it can be quite dangerous. So although oxalate itself can be beneficial if the intake is it's in proper dose. So we have, as a nutritionist, we need to be careful to formulate the diets, including this anti-nutritional contents in the diet, so especially about proper dose. And the last, I have seen the slides from Dr. John Gaughan that sustainable future livestock production system is also affected by climate change. So this climate change can result in livestock is more vulnerable to get physiological heat stress, resulting in reduced production. But Dr. John Gaughan also mentioned in his slides that there are strategies to reduce the disadvantageous effect of heat stress in animal production. I think that's all I can conclude about the topic of this seminar. And again, thank you very much to all invited speakers for your valuable presentation and discussion. Right. Now we are going to the next session, which is closing ceremony. In this session, the committee have appreciation for all the invited speakers, and our dean, Professor Husmi Yurmiati, will chair this session and give appreciation to all the invited speakers. Professor Husmi Yurmiati, are you still with us?
Okay. Thank you, Mr.
Time is yours,
Diki.
please.
Yes. Thank you, Mr. Diki, for much your valuable insight of the future of sustainable livestock production, and thank you for all participants and committee of 57th anniversary of the Faculty of Animal Husbandry. Thank you for Dr. Ahmad Firman. Thank you very much.
Thank you, Bu. Sama-sama, Bu.
And for Dr. Hossam Shawki, thank you for your time in Seminar International. I hope we can extend and maintain our relationship and academic or non-academic program in the future for my faculty, Animal Husbandry, Universitas Padjadjaran. Thank you very much, Hossam Shawki.
Thank you very much for nice. It's my pleasure, and we welcome any collaboration between Nagoya City University and your university in near future.
Okay, thank you. And then Dr. Mijanur Rahman, speaker in International Online Seminar, Sustainable Future Livestock Production System. The same, I hope we can extend and maintain our relationship and in academic and non-academic program in the future. Thank you very much, Mr. Mijanur.
Yeah, thank you, Prof. Thank you very much.
And then for Dr. John Gaughan, and speaker international online seminar, Sustainable Future Livestock Production System. I hope we can extend and maintain our relationship in academic and non-academic program in the future. And I hope you get well soon. Thank you very much, Mr. John. And for Dr. Diki Ramdhani, thank you very much for your valuable insight on the future of sustainable livestock production. Thank you very much, Dr. Diki.
Thank you very much, Professor. You're most welcome, Professor Kusmi Yurniati.
Okay. Please, Dr. Diki.
Thank you very much, Professor Kusmi Yurniati, for chairing the appreciation sessions, including closing ceremony. I think this end of our event, on behalf of the committee, I would like to thank you for all the participants, especially invited speakers, invited institutions, and all the participants. Also, we would like to say sorry for any inconvenience caused during this event. We are sorry for any inconvenience. So we are going to close this event by saying Alhamdulillahirabbil alamin. Good morning and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Can we have a picture first, Dr. Diki?
Yeah, that's good idea, Dr. Nopi. How technically we can do it?
Maybe the upgraders or Patoni can help us to screenshot per screen.
Right.
Allow me to take screenshot. Please open your camera and hold your best position. I will take screenshot. Wait for 1, 2, 3. Next. Hold still. 1, 2, 3. Yes, it's done, Dr. Diki.
Thank you very much, Ms. Tony Pramulyadi, our host and our IT staff in the faculty. So, Dr. Nopi Mayasari,
Thank you so much, everyone. So maybe I hope you enjoy this event and see you in the next event. Okay. Bye.
Dr. Nopi Mayasari,
get well soon for Dr. John Gaughan, and thank you for everyone, especially thank you for the invited speaker.
Thank you, Dr. Nopi.
Okay, thank you, Mrs. Topi. Goodbye.
Bye. Thank you all.
Thank you.
Good morning, I'm Tony Pramulyadi as Faculty Admin for this occasion. I would like to thank you all for participating in today's seminar hosted by Animal Husbandry Faculty of Padjadjaran University. Now allow me to close the virtual room and thank you for using our services. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, panitia. Terima